Science 2 subject. I'm from Mithanagar, Mumbai Public School. We are going to revise 16th chapter of Science 2, that is heredity and variation, today. And also, we are going to solve the MCQ based on this. So, hello.
Yes, teacher. Hello.
Am I audible, sir?
Yes, teacher.
Is my screen visible?
Not yet, teacher.
Presentation?
No.
Education Department welcomes you for state live lectures. I am Yasmeen Sheikh from Mithanagar, Mumbai Public School. And today we are going to conduct a Science 2 lecture. And in this lecture, I am going to revise 16th chapter, that is heredity and variation. And also we are going to solve MCQ based on this. So I would like to request you that you pay attention so that you are able to answer my MCQs. Okay, so we'll do a quick revision. Heredity. What is heredity? Transfer of characteristics from parents to offspring is called heredity, right? So whatever the characters are transferred from parents to offspring, that is called as heredity. It is due to heredity that puppies are similar to dogs and the infants are similar to human beings. This we have already studied, right? Then in this chapter, we have also studied about chromosomes. What are chromosomes? The structure in the nucleus of cells that carries the hereditary characteristics is called the chromosome, right? So it is a carrier of the genetic material. All the genetic material is stored in this chromosome. So chromosome is made up of nucleic acid and proteins. That means chromosome is made up of the DNA and proteins. And also we have studied the structure of chromosome. We know that there are two arms to the chromosome, P-arm and Q-arm, and a centromere is also present. So depending upon the position of the centromere, there are four types of chromosomes: metacentric, submetacentric, acrocentric, and telocentric. What is metacentric? When the centromere is exactly at the midpoint in the chromosome, that is called as metacentric chromosome. It looks like the English letter B, isn't it? Then submetacentric, the centromere is somewhere near the midpoint in the chromosome, and that is why it is called as submetacentric chromosome. The third is acrocentric chromosome. In this type of chromosomes, the centromere is near one end of the chromosome. That is why it is called acrocentric, and it looks like the English letter J. Telocentric, in this centromere is right at the end, that is telo. Telo means the end point of this chromosome, making the chromosome look like the English letter I, right? So chromosomes are the genetic material present in the nucleus, and depending on the presence of centromere, chromosomes are of four types. You should remember their names: metacentric, submetacentric, acrocentric, and telocentric. Then next in this chapter, the important point is DNA, right? So we have, in the lecture, we have studied detailed structure of the DNA molecule. right? We have studied almost everything about DNA, DNA fingerprinting, and Human Genome Project, right? So, what is DNA? The full form of DNA is deoxyribonucleic acid. It was first discovered by Friedrich Mischer in 1869. It is also called as the master molecule. While it is called master molecule, because all the functioning of the cell and the functioning of the body is controlled by this DNA. In 1953, Watson and Crick produced a double helical model of the DNA. These were the people who have found out how the DNA looks. And DNA is made up of a small molecules, right? Each strand in the molecule of DNA is made up of many small molecules known as nucleotides. And I hope everyone knows how the nucleotides are made. Nucleotides are formed of deoxyribose sugar, phosphate molecules, and four types of nitrogenous bases: adenine, guanine, cytosine, and thymine. Right? Then the next point is gene. Segments of the DNA molecule are called genes. So the small segments of the DNA molecule are called as genes. And genes control the structure and the function of the cell and of the body. So this small part, it is responsible for the synthesis of proteins. In whatever particular type of protein will be synthesized, that information is stored on the genes. So it is a gene is nothing but a small part or a small segment of DNA molecule. The next is RNA. The full form of RNA is ribonucleic acid, and it is the second important nucleic acid in the cell. It is also made up of many small nucleotides, and the nucleotides of RNA are made up of ribose sugar. Here, the difference between DNA and the RNA is DNA is made up of deoxyribose sugar, while RNA is made up of ribose sugar. Then phosphate molecules are there, and also there are four types of nitrogenous bases, that is adenine, guanine, cytosine, and uracil. And again, one more difference between DNA and RNA is the uracil is a nitrogenous base which is present in RNA, but it is not present in DNA. Okay. Then, so depending on the function, there are three types of ribosomes, that is rRNA, mRNA, and tRNA. So rRNA stands for ribosomal RNA, mRNA means messenger RNA, and tRNA means transfer RNA. So first I'll explain you messenger RNA, the RNA molecule that carries the information of protein synthesis from genes, that is from DNA, in the cell nucleus to ribosomes in the cytoplasm, which produce the proteins is called as messenger RNA, right? So messenger, from the name itself, it conveys that this is a messenger RNA which carries the message from DNA to ribosomes to synthesize the proteins. Then the next is the transfer RNA, the RNA molecule which according to the message of the mRNA carries the amino acid up to the ribosomes is called as transfer RNA. Then with the help of the information brought by messenger RNA, so what kind of amino acids are required for protein synthesis? Those amino acids are carried by transfer RNA to the ribosome where the actual synthesis of protein is going to take place. Then rRNA, the molecule of RNA, which is a component of the ribosome, right? So this rRNA, ribosomal RNA, is present in ribosomes, and that is why it is called as ribosomal RNA. And these ribosomes perform the actual function of protein synthesis, right? So the information is carried by messenger RNA, then Amino acids are provided by transfer RNA, and the actual synthesis takes place in ribosomes with the help of ribosomal RNA. Then next in the chapter is Mendels Principles of Heredity. Now we have studied in details about Mendel. Victor Johann Mendel was an Austrian scientist, and he studied the inheritance of the characteristics from the pea plant. We know that all of the Mendel's experiments were based upon the visible characteristics of the pea plant. So in this chapter, we have studied the genetic material is transferred in equal quantity from parents to progeny, right? We all know that the genetic material is carried in equal quantity from both the parents, that is father as well as mother, or you can say in case of animals, male parent and the female parent. So or in plants also. So the genetic material is transferred in equal quantity from parents to progeny. So the principle of heredity is based upon this fact. Now he has studied that if equal quantity of genetic material is transferred, then which character will appear in the progeny? So this he has studied with the help of pea plants. So we have studied Mendel's monohybrid cross and Mendel's dihybrid cross. Right. So the example given in the textbook and which we have studied is the cross between pure tall plant and pure dwarf plants. Right. So we have seen that when the pure tall plant was crossed with pure dwarf plant, then a hybrid tall plant was found, and that is called as the first serial generation. And then when the selfing was taken place between first serial generation, then the it was solved with the help of Punnett square, right? And then you can see okay, I am trying to show you with the help of a spotlight or okay, I found it. So here it is. So selfing is done. And you can see here, this table is called as the Punnett square, and we have seen how to write male gametes and female gametes and how to do the cross. So what is important in this is, so in Mendel's monohybrid cross, he has found is that the phenotypic ratio of the Mendel's monohybrid cross is 3 is to 1, 3 tall plant is to 1 dwarf plant, while the genotypic ratio is 1 is to 2 is to 1. That is 1 pure, 2 hybrid, and 1 pure again. Right? So, in this Mendel's monohybrid cross and dihybrid cross, both are very important topic, students, and you practice to solve the crosses, and you have to remember their phenotypic and genotypic ratios. Then the next is Mendel's dihybrid cross. So in this, we have studied the cross was done between rounded yellow seeds and the wrinkled green seeds. For that, we have taken the genotype capital R, capital R, capital Y, while for the wrinkled green seeds, that is recessive character, we have taken small r, and small y, right? So with that information, we have solved the Mendel's dihybrid cross, and what we have found is the phenotypic ratio was 9 is to 3 is to 3 is to 1, while the genotypic ratio was very wrong, right? 10, the next topic in this chapter is genetic disorders. Diseases or disorders occurring due to abnormalities in the chromosomes and mutations in the genes are called genetic disorders, right? As we all know, if the disease is because of virus, that we call it as viral disease. If disease is because of bacteria, we call it as bacterial disease. In the same way, here the diseases occur due to abnormalities in the chromosomes, and now we know that in human beings, we have 46 chromosomes, or in other words, we can say there are 23 pairs of chromosomes present, right? So, if there is any abnormality in the chromosome, so or any mutation of the gene, so that leads to genetic disorders, right? So, the example of the genetic disorders given here are Down syndrome. We have studied in details about all these diseases. Down syndrome. So I'll tell you in short, Down syndrome is a disorder arising due to chromosomal abnormality. And this was the first discovered chromosomal disorder. And in this Down syndrome, it is described as trisomy of the 21st chromosome, right? So normally the chromosomes are present in pairs. We say that there are 23 pairs of chromosomes, but on the 21st pair of chromosome in each cell, there is an extra chromosome present. That is 46 plus 1 condition, or it is also called as trisomy of the 21st chromosome. That means instead of two, three chromosomes are present in the 21st pair, right? And that is why this causes the Down syndrome. In Down syndrome, the symptoms of the Down syndrome are the children will have short height, they have short wide neck, flat nose, right? And their mental retardation is the prominent characteristic. So the children or the people who are suffering from Down syndrome, they're mentally retarded, you can say, and their life expectancy is also less, right? Then the next is Turner's syndrome. So here you can see monosomy of the X chromosome in Turner syndrome. So most of this time we know that there are 22 pairs of autosomes and one pair of sex chromosomes, right? It can be either male sex, that is XY chromosome, or it can be a female sex chromosome, that is XX chromosome. So it should be either 44 plus XX or 44 plus XY. But in this case, because as with autosomes, abnormality in the sex chromosomes also takes place. So in Turner's syndrome, due to inactivation of one of the X chromosomes, so it looks only 44 plus X condition, right? So instead of 44 plus XX chromosome, it is 44 plus X. That means women suffering from Turner syndrome show 44 plus X condition, and such women are sterile, so that is called as Turner's syndrome. Then the next is the Klinefelter syndrome. This Klinefelter syndrome, this is the disorder arises in men due to abnormalities in the sex chromosome. Again, in this disorder, men have an extra X chromosome, hence their chromosomal condition becomes 44 plus XXY. If they are men, what should be their chromosomal condition? 44 plus XY. But in this case, it becomes 44 plus XXY. One extra X chromosome is there due to some abnormality in the chromosome, and which leads to Klinefelter syndrome. So these are all genetic disorders: Down syndrome, Turner syndrome, Klinefelter syndrome. Also, diseases occurring due to mutations in the single gene. We know that DNA is made up of genes, right? A segment of the DNA is called as gene. So the whole of the DNA is made up of many genes. So if there is mutation in one single gene, that kind of diseases are called as monogenic disorders. The example given in our textbook is albinism, sickle cell anemia. Also, there are many different examples: Hutchinson's disease, galactosomia, phenylketonuria, sickle cell anemia, cystic fibrosis, hemophilia, night blindness, etc. are the examples of monogenic disorders, right? That means in this case of in such diseases, mutation is there in the single gene. Now, albinism, you all know, it is a genetic disorder. Our eyes, skin, hair, everything becomes colorless. That is, it is without the presence of melanin pigment. Then the next type of diseases are disorders due to mutations in multiple genes. That is polygenic disorders, right? So instead of one, if there are mutations or changes in many genes of the DNA, more than one, you can say more than one gene of the DNA, that is called as polygenic disorders. And again, there are many different examples of polygenic disorders: cleft lip, constricted stomach, diabetes, asthma, blood pressure, heart disorders, obesity, such kind of many different diseases are there, which are the example of polygenic disorders. Okay, so that means genetic diseases are classified into four types, that is disorders due to chromosomal abnormality, diseases occurring due to mutation in single gene, diseases occurring due to mutation in many genes, and also one more type is that is mitochondrial disorder. So you know that DNA is also present in mitochondria. Mitochondria is one of the cell organelles, and mitochondrial DNA may also become defective due to mutation, right? And this disease, that is liver hereditary optic neuropathy, this is also an example of mitochondrial disorder. And the next is polygenic disorder, right? Now we'll move on to the next topic, the last topic of the chapter, but which is very important. I would like to emphasize on this. I would like to explain you interrelationship between tobacco addiction and cancer, right? So you must have seen that it is written on all the tobacco products that tobacco is injurious to health. It may also cause death, right? So let us see what is the reason. Consumption of tobacco in any form can cause cancer. Tobacco consumption can also lead to shortening of the lifespan, chronic bronchitis, pericarditis, cancer of the lungs, mouth, larynx, pharynx, pancreas, urinary bladder, etc. Right? So you can imagine how dangerous this tobacco, causing so many different types of cancers all over the body. Right? And this is because harmful effects of the smoking are due to the nicotine present in the tobacco. Also, you know that smoking is a kind of taking tobacco inside, because the cigarettes, they consist of tobacco, and tobacco consists of a chemical that is called as nicotine, right, which is very dangerous. And also, I would like to tell you about the cancer. Cancer is nothing but the uncontrolled growth of the cells. You know, this nicotine or whatever different types of chemical, harmful chemicals which are present in the tobacco, they mutate your DNA, right? That means they bring abnormality in your DNA, so that the cells of the body starts growing uncontrollable. That means uncontrolled growth of the cells takes place because of mutations in the DNA. So you can see that this tobacco is directly affecting your DNA, and once there is a problem, or once there is when something is wrong with the DNA, you know that most of the diseases are curable, but if there is any damage to the DNA, then it is very difficult to cure. So that is why this tobacco, it directly affects your DNA. Some kind of mutations takes place because this cancer is not a viral disease. Cancer is not a bacterial disease. Cancer is something that means some chemicals are there which are affecting our body, which are directly affecting the DNA of our body, and which is bringing about such kind of diseases, that is uncontrollable growth of the cells, right, in any part of the body, sorry, maybe in mouth, or in neck, or stomach, intestine, or any other part. So the other reasons are also there for cancer, but they are very rare, right? That means, for example, I'll tell you, for example, the chemicals or the pesticides which are used on the fruits or vegetables, or the food grains what we are eating, if they are if much use of chemicals are there, so if we are by any way, if it is going in our body, so it may mutate our genes, it may mutate our DNA, and the cancer can be caused. But the 90% of the reason of people suffering from cancer is this tobacco. And tobacco smoke contains harmful chemicals like pyridine, ammonia, aldehyde sulfuryl, carbon monoxide, nicotine, sulfur dioxide, etc. So, whatever these names you are hearing, these are all poisonous gases which are very harmful for us. They cause uncontrolled cell division, right? So, why the cell is dividing uncontrollably? Because somebody is giving information for the cell to divide it. And who gives information to cell? Who is the controller of the cell? It is DNA. That means some kind of mutation is going on in the DNA because of this taking tobacco. So here you can see the whole of this is oral cancer. The whole mouth is affected. So it is written, smoking causes 92% of the oral cancers. Here you can see healthy lungs and the smoker's lungs. It turns black because of the smoke, right? So I would like to request you, I would like to kindly please request the students that any kind of intake of the tobacco, it can be in the form of gutkhas, it can be cigarette, it can be bidi, and many different types are there, which are available in the market. So now, being responsible or being educated, it is our responsibility that we should not touch any tobacco product, right? And also, we should inform others the harmful effect of the tobacco, maybe in our family or in our surrounding, in our friends. We should try to make them understand that eating tobacco, that means it is affecting our life, or maybe decreasing our life. And the cancer is such a danger disease, or that means it is very painful. So I request all the boys and girls to be away from any tobacco product, even if somebody is giving you in free, right? So in the same way, the others also, maybe other different kinds of drugs. So please don't. So because this cancer is, if you meet any cancer patient, you will find that in how much pain they are, right? So I hope you understand. So to protect oneself, so here is the most important line. To protect one's body from cancer, one must avoid smoking and consumption of tobacco and tobacco products in any form. right? And also, passive smoking is also there. That means if some person is smoking, and if you're standing there near that person, and if that smoke is going into your nose and your body, and that is just like smoking. Even if you're not smoking, but the smoke is going in your body, that means that nicotine and other different harmful gases are going inside your body. So you should protect that also. You should see that you don't stand near any such person who is smoking. Okay, I hope you have understood the chapter. This was revision of heredity and variation. Okay, so we will stop here, and also now I will give you one multiple choice question test, and I want that all of you should respond. You can one by one raise your hands for the answers. I will share you the questions. So here is a small multiple choice MCQ, that is multiple choice questions, which I would like you to answer. Okay. Sir, please unmute students,
Okay, teacher.
and those who wish to answer, ask them to raise their hand. I don't know, I can't see participants. So is there any option that I can see participants on the screen? No, I am not able to see the participants. Sir, you please ask them, those who are
Yes,
raising their
teacher.
hands, to answer.
Yes. Yes, teacher. Sure.
Okay, so the first question is, Gregor Johann Mendel was an dash scientist. The options are Austrian, Swiss, German, Indian. Those who
Yes,
wish to
Sakshi.
answer,
Australian. Austrian.
yes, it's Austrian. Very good. It's not Australian, it's Austrian. Right?
Yes.
That is the correct answer. Very good. Okay, so I've marked the answer, what Sakshi has told. Now we will move on to the next question. In Mendel's experiment, pure dwarf plants are denoted by genotype dash.
Yes, Krish.